At the lip of a burrow in shallow tropical water, resting on pale sand no more than a few meters below the surface, a creature roughly the length of a human thumb holds very still. The water above it is warm and filtered, and where the light comes through, it catches the translucent joint of one oversized claw, pale against the animal's darker body, held slightly outward from its side, half open. The creature is a pistol shrimp, and for something so small, the world it occupies is surprisingly complete. The family it belongs to is called Alphidae, and it is a large one. More than 1,100 species have been formally described within the group, which places it comfortably among the major crustacean families, related through the broader order Decapoda to crabs, lobsters, and the barnacles clinging to rock faces in the intertidal shallows above. That company might seem grand for an animal that fits without difficulty in a closed hand, but taxonomy follows its own sense of proportion. The pistol shrimp belongs where it belongs, and its family has been producing variations on the same basic body plan for longer than most of the reef structures it inhabits have existed. Most pistol shrimp measure somewhere between 3 and 5 centimeters from the tip of the rostrum at the front of the head to the fan of the tail at the back. That is the range for a typical adult, the kind of animal that would rest if placed in a palm, without much room to spare on either side. Their coloration varies considerably, depending on where they live and which species they are. Species that occupy mudflats and sandy seafloors tend toward the colors of those environments: olive green, sandy brown, muted yellow, colors that make the animal's edges difficult to separate from the background when the light is low. Species associated with coral reefs often carry more vivid markings: vivid orange, deep red, purple banding across the legs and body, as though the reef's own palette had been applied to them. Some carry both, the body richly colored and the large snapping claw paler and more translucent, almost as though the claw was added later and hasn't quite taken on the same pigment as the rest. That claw is the first thing most people notice when they look closely at a pistol shrimp, and it is easy to understand why. In some species, it constitutes nearly half the animal's total body weight. A 5-centimeter shrimp with a claw that accounts for that proportion of its mass is carrying something genuinely disproportionate, something that looks, to an observer expecting the paired, roughly equal chelipeds of most crustaceans, like a mistake in the body plan, or a feature borrowed from a much larger animal and fitted to a smaller frame. It is neither of those things. The claw is exactly what the animal has evolved to have, shaped over enormous time into its present form, and the asymmetry is structural rather than accidental. The pistol shrimp snapping claw operates through a mechanism that, described in purely physical terms, involves a plunger and a socket. One half of the claw, the dactylus, has a rounded projection at its base, the plunger. The other half, the propus, has a corresponding hollow, the socket. When the claw is cocked open and then released, the dactylus snaps closed, and the plunger drives with considerable force into the socket. And it is from this interaction, this extremely rapid, extremely precise closure, that everything else the claw does follows. The claw in its resting position, held slightly open, slightly outward from the body, is a loaded mechanism. Even at rest, it carries the tension of a body that has learned, over millions of years, how to store and release energy very quickly. The other claw, the one on the opposite side of the body, is small and ordinary, and is used for the things most crustacean claws are used for: grooming, handling food, occasional light defense. It is the smaller claw that makes the large one look so unusual. Seen together, the two chelipeds create the impression of an animal that grew up asymmetrically, investing almost everything on one side and leaving the other to manage on its own. Beneath the snapping claw, and below the two walking legs that extend from the same side of the thorax, the body of the pistol shrimp tapers into a relatively standard crustacean arrangement: a segmented abdomen, a uropod-flanked telson at the tail, and pleopods along the underside of the abdomen that serve in locomotion and, for females, as a surface for carrying eggs. A pistol shrimp moving across the seafloor moves in the way of most small shrimp, in bursts, sometimes walking on its periopods across a surface, sometimes swimming backward with rapid abdominal flexion when a faster movement is needed. Most of its life, though, is spent close to the burrow, at the entrance just outside it, or making short foraging excursions into the nearby substrate before returning. The head of a pistol shrimp carries two pairs of antennae, and together they are among the most important structures on the animal's body. The longer pair, the antennae proper, extend forward and to the sides, reaching out a distance roughly comparable to the animal's own body length. They are fine and flexible, and they move in slow sweeping arcs, reading the movement of the water around the animal. When something passes close enough to disturb the water, a fish, a shadow, a change in the current, those long antennae register the disturbance before anything else does. They are, in this sense, a kind of long-range early warning system, though the warning they issue is felt more as a shift in the quality of the water than as anything like a sound or an image. The shorter pair, the antennules, are positioned closer to the front of the head and are dense with chemoreceptors. They read the water not for movement, but for chemistry, sampling the dissolved molecules drifting past the animal to identify what is near: food, a potential mate, the specific chemical signature of a symbiotic partner, the boundaries of its own territory as marked in the substrate. Pistol shrimp live in a world that is, for them, primarily a world of chemical texture. The water they inhabit is never chemically neutral. It is full of information, and the antennules are the instrument through which that information arrives. The compound eyes of a pistol shrimp are mounted on short, mobile stalks at the front of the carapace. They move, they register light and motion, and they contribute to the animal's ability to navigate its environment. But they are not the primary organ through which the animal understands its world. Pistol shrimp that have lost an eye, or that live in environments where the light is too dim for vision to be useful, seem to manage their daily lives with very little apparent disruption. The antennae, by contrast, are consulted constantly. Their absence or damage produces a measurably more disoriented animal. The eyes are useful. The antennae are essential. Pistol shrimp inhabit a wide band of the world's oceans. They are found wherever the water is warm enough and the coastal structure is suitable: tropical and subtropical regions across the Indo-Pacific, the Caribbean, the Gulf of Mexico, the eastern and western Atlantic, the Red Sea, the coasts of northern Australia, and the Indian Ocean rim. The specific environments they occupy within those regions vary: coral reefs with their complex rubble and overhangs, seagrass beds where the sediment is soft and fine, and the canopy of grass blades provides partial cover from above, mudflats at the edges of mangrove systems, sandy seafloors in shallow lagoons where the water is clear and the light reaches the bottom in long shifting columns through the surface. What most of these environments share is a depth range that keeps the animal within reach of the light, typically somewhere between the intertidal zone, where the water recedes at low tide, and approximately 40 meters below the surface. Very deep water, with its cold temperatures and high pressure, is not pistol shrimp territory. They are animals of the shallow, warm, lit places. In these places, the pistol shrimp is rarely a conspicuous presence to a casual observer. It is too small and too given to remaining close to its burrow to stand out in the busy visual environment of a coral reef or a seagrass bed. A diver swimming above a reef might pass over hundreds of them without registering any individual animal, might register only the crackling texture of the ambient sound coming from the sediment below, not knowing yet what is making it. The shrimp, for its part, is probably aware of the diver long before the diver is aware of the shrimp. The bow wave of the diver's fins, read through those long sweeping antennae, would arrive well before the diver's shadow. The burrow is the center of the pistol shrimp's world. Not every species of pistol shrimp builds one. There are members of the family that live in sponges, or in rubble, or in the spaces between coral branches. But for the species most people mean, when they say pistol shrimp, the ones found in sandy seafloors and seagrass beds and reef rubble, the burrow is both home and project. The animal constructs it, maintains it, defends it, rests in it, reproduces in it, and in some cases spends nearly its entire adult life within a few centimeters of its entrance. The burrow is not simply a shelter. It is the shrimp's primary contribution to the physical structure of the seafloor it inhabits, and the digging and maintenance of it takes up a substantial portion of the animal's active time. The entrance of a pistol shrimp burrow is easy to overlook. It is typically a small round or oval opening in the sediment, a centimeter or two across, with a low mound of excavated material piled just outside it, grains of sand or silt, or small fragments of shell that the shrimp has carried from inside the burrow and deposited at the threshold. That mound, small as it is evidence of ongoing work. The shrimp keeps the burrow clean, removes material that falls in, adjusts the entrance when the substrate shifts. The mound outside the entrance is never quite the same from one day to the next, because the work that produced it is never quite finished. At shallow tropical reefs, where reef rubble and broken coral fragments are mixed into the sandy substrate, the entrance to a pistol shrimp burrow is sometimes framed by pieces of rubble that the shrimp has placed or gathered, not random debris, but material that functions as a kind of structural reinforcement for the opening, keeping the entrance from collapsing when the water moves. A pistol shrimp with a well-maintained burrow entrance may be an animal that has lived in that location for some time, incrementally refining a structure it first constructed days or weeks or months ago, carrying one grain at a time. The coloration of reef-associated pistol shrimp sometimes makes them briefly visible at the entrance of their burrows during the parts of the day when they are most active. A vivid orange shrimp with deep red or purple banding on its legs, resting at the lip of its burrow in the rubble of a shallow reef, is not a difficult thing to see, but it is still a thing that requires knowing where to look, and that requires the patience to hold one's attention on a small area of substrate long enough for the shrimp to reappear after a disturbance has sent it retreating inside. The color that seems conspicuous against the white or gray of a coral fragment matches well enough against the rust-colored rubble and encrusting algae of a living reef that the animal becomes part of the texture of the place rather than a feature standing out from it. Inside the burrow, the structure is more complex than the modest entrance suggests. The tunnels extend down into the substrate, angling and branching in ways that the simple opening gives no hint of. There may be a section where the animal retreats during rest, a section where it processes food, a section in a female shrimp carrying eggs, where she tends the small mass of developing embryos attached to the underside of her abdomen. The shrimp that built the burrow knows its structure the way a creature that has spent months in a particular space knows it, by feel, by chemical trace, by the accumulated experience of having moved through it thousands of times. It does not need to see in the dark interior. The antennules read the walls, the legs feel the floor. The burrow is a known space in a way that the open water above it never quite is. A pistol shrimp that encounters open water, that is water without the immediate proximity of a burrow wall or the substrate of the seafloor, will typically move quickly to re-establish contact with the surface. The open water column is exposure, the burrow is security. Between those two states is the foraging zone, just outside the entrance, the small radius of the seafloor that the shrimp moves across during its active periods, antennae extended, claw held outward, working methodically across a known space before returning. That radius can be quite small. In some species and some habitats, the foraging territory extends only a few centimeters from the burrow entrance. In others, the shrimp ranges somewhat further, but rarely far, and rarely for long, and rarely without periodic retreats back to the entrance to deposit material or briefly rest. The world as the pistol shrimp experiences it, then is a world organized around the burrow. Distance is measured from the entrance. Safety is measured by proximity to it. The chemical landscape of the surrounding substrate, what has moved through recently, what has settled in the sediment, what the water column has carried past, is processed constantly through those dense chemoreceptive antennules, building a picture of the local environment that is precise and continuously updated. It is a different kind of spatial awareness than vision provides, but it is not a lesser one. It is for this animal in this environment, the appropriate one. The sediment itself is not neutral to a pistol shrimp. Sandy seafloors and seagrass beds contain an enormous variety of chemical signals, organic material decaying, small organisms living and moving through the substrate, the traces left by other animals passing above. A pistol shrimp processing all of this through its antennules while it forages is doing something that looks, from a distance, like simple wandering, but is in fact continuous active sampling of an information-rich environment. When the antennules pause and press toward the substrate, when the animal holds still for a moment before moving again, it is often attending to something chemical in the sediment that has drawn its attention. The compound eyes on their mobile stalks are scanning during this time. Movement registered by the eye, a shadow passing, a fast-moving shape at the edge of visual range, can trigger a rapid response, even in an animal whose primary sensory investment is chemical rather than visual. The eyes and the antennae work in parallel, each contributing a different kind of information to the animal's moment-by-moment understanding of its surroundings. The world the pistol shrimp inhabits is not dark or featureless, even in the dimmer sections of the reef, in the shadows between coral heads, where a burrow is often constructed. There is enough light for the compound eyes to register the broad shapes of nearby objects and the movement of the water above. A pistol shrimp that lives long enough will spend 3 to 4 years in this way. That is the estimated lifespan for the species in the wild. Not a long time by many measures, but not a brief one either, for an animal of this size. 3 to 4 years of burrow maintenance, of foraging across a small radius of familiar substrate, of processing the chemical texture of the water and the mechanical texture of the current through those long antennae. 3 to 4 years of the compound eyes, reading the shifting light from above. 3 to 4 years of carrying and tending eggs through breeding cycles, of knowing one particular patch of sandy seafloor and one particular tunnel in the sediment with the accumulated precision of many months of continuous habitation. The shallow tropical waters that pistol shrimp inhabit are not quiet places, though they might appear so from a distance. They are full of movement and chemistry, and the constant small transactions of a densely populated ecosystem. Fish pass above, invertebrates move through the substrate. The water column carries chemical information from far away. The light changes with the angle of the sun and the movement of the water surface above. The pistol shrimp, sitting at the entrance of its burrow, with its long antennas sweeping and its chemoreceptive antennules reading the water, is attending to all of this in the only way it can, continuously, from a fixed point, through the specific sensory equipment it carries. In a seagrass bed, the burrow entrance sits among the roots and rhizomes of the grass, which stabilize the sediment and provide additional structure to the shrimp's immediate environment. The mound of excavated material outside the entrance sits at the base of a grass stem or in a small clearing between the rhizomes. Above the entrance, the grass blades move gently with the water. The water is warm and clear in the shallow tropical lagoon. Light reaches the seafloor in broad, shifting columns through the moving surface above. A pistol shrimp resting at its burrow entrance in a seagrass bed is a very small thing in a very specific place, and the place, if one keeps still enough and looks at it carefully enough, turns out to be layered and complex in ways the first glance doesn't reveal. At a mudflat on the edge of a mangrove system, the environment is different in texture and color, but similar in its organization around the shrimp's specific patch of substrate. The water here is often turbid, carrying fine particles of organic material suspended through the water column, and the seafloor is soft and dark. The pistol shrimp's olive or brown coloration matches the substrate almost exactly. Its burrow entrance is a small opening in the mud, and the mound of excavated material is dark and fine-grained and barely distinguishable from the surrounding sediment. The animal foraging across the surface is nearly invisible, not because it is hiding, exactly, but because it is the color of the place it has chosen to live. On a sandy seafloor in a shallow tropical lagoon, the pistol shrimp's pale sandy coloration performs the same function. The snapping claw, lighter and more translucent than the rest of the body, catches the light from above in a way that the rest of the animal doesn't quite. And this pale glinting is sometimes the first thing that reveals the shrimp's presence to an observer who has been watching the substrate carefully. The pale joint of the claw, illuminated from above, holds the light for a moment as the shrimp shifts position on the sand. Then the animal moves, the angle changes, and the light no longer catches it in the same way, and the shrimp becomes sand again. What exists inside that claw, what the pale translucent structure is capable of, is not yet part of this picture. The claw here is simply a structure, a feature of the body, something the animal was born with in miniature and grew into over the months of its first year, something it carries with it the way it carries every other part of itself, constantly, without awareness of its own strangeness, as simply part of being what it is. The claw is held slightly outward, in the way that a claw of that size and weight naturally comes to rest when the animal is at ease. It is part of the animal's resting posture. It is not, at this moment, doing anything more than being there, pale and translucent and specific, at the edge of a burrow entrance on a sandy seafloor in warm, shallow, filtered tropical water. The pistol shrimp as a body, then, small enough to rest in a closed fist, heavy on one side with a claw that constitutes nearly half its total weight, colored to match the substrate of its preferred habitat, navigating by chemistry and by the felt movement of the water through long and constantly moving antennae. Living within a short distance of a burrow, it has built and tends and knows, in fine detail, occupying a lifespan that will run to perhaps 3 or 4 years before it is finished. A small animal, a specific one, an animal with a particular family, a particular place in the classification of living things, a particular set of sensory priorities that have organized its body and its behavior in ways that make complete sense for the life it lives. The water above the shallow tropical lagoon is warm and very clear. The light that comes through it falls on the sandy bottom in slow shifting shapes, and in the bottom the small burrow entrance sits with its mound of excavated material outside it, and at the entrance the shrimp rests, and the long antennae sweep slowly through the water, and the compound eyes on their short stalks read the light and the shadow of everything passing above. Somewhere in the sediment below, the tunnel extends its full 20 or 30 centimeters into the seafloor, turning and branching through the darkness, a structure the animal built grain by grain over the months of its life in this location. The shrimp has lived here for some portion of what will turn out to be a 3 to 4 year existence. It will go on living here, on this particular patch of sandy seafloor, maintaining this particular burrow, foraging across this particular small radius of substrate for whatever remains of that time. The world it occupies is not a large one, measured in the way a larger animal might measure space, but it is a complete one, and it is on this quiet afternoon in the shallow water above the sandy seafloor, very precisely occupied. The two halves of the snapping claw come together at roughly 100 kilometers per hour. That single fact takes a moment to settle, because the animal producing it is smaller than a human thumb, and the event it describes is over before the word asterisk snap. Asterisk has finished forming. The claw closes. A jet of water shoots outward from the point of closure, moving fast enough that the pressure in the water immediately behind it cannot fill in quickly enough. The water in that narrow wake briefly ceases to behave like water. Its local pressure drops below the threshold at which it can remain liquid at the surrounding temperature, and for an instant, a fraction of an instant, the water there becomes vapor, a bubble forms, it expands, and then, because the pressure differential that created it is already equalizing, it collapses inward on itself, with a force that has very little in common with the quiet coastal seafloor in which it occurs. The bubble is gone before most biological nervous systems could register that it had appeared. It exists for approximately 700 microseconds, 7/10,000ths of a second, and in that interval, it opens and closes a chapter of physics that most people associate with industrial machinery rather than with a crustacean foraging in the sediment of a tropical reef flat. This kind of bubble, formed when fast-moving liquid creates a local pressure low enough to vaporize itself, is called a cavitation bubble. The word comes from the Latin for hollow. What it describes in the case of the pistol shrimp is something considerably stranger than its name suggests. The sound produced when that bubble collapses has been measured at up to 210 decibels underwater. That number is large enough that it requires a brief frame to make it felt. A jackhammer breaking concrete at close range produces roughly 100 decibels in air. A jet engine at takeoff distance produces roughly 140. The scale is logarithmic, which means each 10-unit increase represents a tenfold increase in intensity, and the distance between 100 and 210 is not a matter of degrees, but of orders of magnitude. Underwater acoustics and airborne acoustics are not directly comparable because the medium transmits pressure differently, but the ratio still conveys something real. The sound of a pistol shrimp snapping its claw is among the loudest biological sounds produced by any animal of its size, anywhere on the planet, and it is produced not by the claw contacting anything, not by any kind of physical percussion, but by the collapse of a small pocket of water vapor in the water beside the claw tip. The claw itself, when it fires, does not strike prey. Nothing is hit. The mechanism is subtler than that, and in some ways more precise. What travels outward from the collapsing bubble is a shock wave, a rapid outward pulse of pressure that moves through the surrounding water faster than the small fish or crustacean nearby can respond to it. The shrimp's target, if there is one, does not see the claw move and has no time to process what the claw's movement might mean. The shock wave arrives first. For an animal within a few centimeters of the snap, the experience, if that word can be used here, is not a strike from a visible threat, but a sudden pressure event with no traceable origin, followed by stillness or disorientation, or depending on the proximity, an end to whatever it had been doing. The shrimp then moves in. The claw never made contact. The work was done by the water. It is worth sitting with that sequence for a moment. The pistol shrimp does not bite, or sting, or chase, or inject. It produces a specific physical event in the water around its body, and that event, a bubble forming and collapsing in a fraction of a millisecond, does the rest. The shrimp is, in a real sense, not attacking with its claw, but with water. The claw is the instrument that creates the conditions. The water is what carries the force outward. This distinction is not merely semantic. It reflects something genuinely unusual about how energy moves through the shrimp's world, and it places the pistol shrimp in a category of organism that is easier to describe once the physical mechanism is understood than it is to describe from the outside, from the level of behavior alone. The claw that produces this event operates through a mechanism that takes a moment to picture. The two halves of the snapping claw are not symmetrical. One half, the larger and more mobile piece, has a projection, sometimes described as a plunger, that fits precisely into a corresponding socket in the fixed lower half. When the claw opens to its cocked position, muscular tension builds across that joint. When the muscle releases, the plunger half snaps downward into the socket at the speed that has already been described, and the water trapped between the closing faces is expelled as a jet. What makes the mechanism notable, beyond its speed, is its specificity. The plunger and socket create a geometrically controlled ejection. The jet is not a random dispersal of water, but a directed column traveling in a predictable direction. The shrimp can angle its claw. It can adjust where the jet will go. This matters because what the shrimp is aiming is not the claw, but the bubble, and the bubble, if well positioned, will collapse at precisely the distance and angle needed to deliver its shock wave to whatever the shrimp is hunting. For a long time, the loudness of the snap was noticed and recorded without any particular explanation for what was producing the sound. It was assumed, reasonably enough, that the noise came from the claw halves physically colliding, a simple mechanical impact between two rigid biological structures closing rapidly. That assumption held for decades. It was only when high-speed imaging technology developed to a point where the microsecond scale events around the claw tip could be captured that researchers were able to watch in slowed-down footage the appearance and collapse of the cavitation bubble, occurring slightly after the claw had already closed. The claw contact produced a sound of its own, smaller and higher. The cavitation collapse produced the loud one. The two events were distinct and sequential, and the louder of them had nothing to do with impact. That realization reshaped the understanding of what the pistol shrimp was actually doing, and it also raised a question that the same imaging data helped to answer. If the bubble collapsed with enough force to produce a pressure wave capable of stunning prey, what was happening inside the bubble at the moment of collapse. Pressure and temperature are related. When a gas, or in this case a vapor, is compressed very rapidly, its temperature rises. The more rapid the compression, the more extreme the temperature spike. The cavitation bubble does not just collapse, it is compressed from all sides simultaneously by the surrounding water rushing back in. At the moment the bubble reaches its minimum volume, just before it finally disappears, the temperature inside it spikes to a level that took several years after the bubble's roll in the snap was confirmed to actually measure. The estimates that emerged from those measurements are still among the more striking numbers in the biology of small organisms. At the peak of bubble collapse, the temperature inside the bubble briefly reaches somewhere between 4,700 and 8,000 Kelvin. The range reflects real uncertainty in the measurements and the models used to interpret them, but even the lower end of that range is a temperature approached only in particular industrial processes, in the coronas of certain stars, and at the visible surface of the Sun. The surface of the Sun runs at roughly 5,800 Kelvin. The interior of a collapsing cavitation bubble, beside the claw tip of a small crustacean, briefly reaches temperatures in the same approximate range. The duration of that peak temperature is in the nanoseconds, billionths of a second. The bubble at that scale of time is not holding steady. It is already rebounding, already cooling, already dissipating. But for the duration that the conditions exist, they are real, and they are extraordinary, and they occur repeatedly across a shallow tropical seafloor wherever pistol shrimp are snapping. Temperature at those levels, even for nanoseconds, produces light. This is not a metaphor or an approximation. It is a physical consequence of the same thermodynamic processes that make hot things glow. When the temperature inside the bubble peaks, a faint flash of light is emitted. The flash is so brief and so faint that it cannot be seen by the human eye under any conditions, and detecting it required instruments of a specific and rather particular kind. In 2001, researchers Detlef Losa, Barbara Schmitz, and Michel Versluis published a study in the journal Science, confirming that the flash was real. The instruments they used were photomultiplier tubes, devices designed to detect single photons, the smallest discrete units of light, in a darkened laboratory with a shrimp in a tank and the photomultiplier positioned to catch whatever the snap emitted. The instruments registered light, not a beam of it, not a glow visible to any camera available at the time, but light, enough photons to constitute a measurable signal. The researchers named this emission shrimp luminescence. The name carries a faint air of the playful, and perhaps it is meant to. It sits alongside triboluminescence and sonoluminescence in a category of light-producing physical events that operate at scales and speeds well outside ordinary perception, and the prefix grounds it in the animal responsible. Shrimpoluminescence is, by the standards of luminescence generally, an unremarkable amount of light. It is not bioluminescence in the sense of the glowing deep-sea creatures that produce light through biochemical processes. It is thermoluminescence, or something close to it, the light that extreme temperature alone, however briefly sustained, is sufficient to produce. The shrimp does not make this light intentionally, and there is no evidence that it serves any biological function for the shrimp. It is a byproduct. It is what happens to water vapor compressed rapidly enough that it happens inside a living animal, and that it required single photon detection to confirm, makes it the kind of fact that seems like it should have a longer explanation than it does. The photomultiplier tube that registered the flash sits as an image somewhere between laboratory equipment and philosophical instrument, a device built specifically to notice things that are there, but that no unassisted sense could verify. The experiment described in that 2001 paper required a darkened room, careful shielding of ambient light, a shrimp willing to snap on demand, and a detector sensitive enough to count the individual particles that the snap produced. What it confirmed was that for the duration of a few nanoseconds, beside the snapping claw of a small crustacean in a shallow water tank in a laboratory, there existed a physical event producing temperatures and light on the scale of stellar surfaces. The confirmation arrived in a darkened room, registered by an instrument, and then recorded on paper in the form of a graph showing a small peak at the moment of the bubble's collapse. The graph is, in a sense, a portrait of something that happened faster than anything biological, inside something smaller than a fingernail, at a temperature that persists for less time than it takes a synapse to fire. A pistol shrimp hunting operates, then within a set of physical events whose character is not immediately obvious from the behavior visible at the animal's scale. From a distance, from the perspective of a researcher watching through an underwater camera, the shrimp moves, pauses, angles its claw incrementally, and snaps. The motion takes a moment. The snap is audible. A small creature nearby may go still, but compressed into the interval between the claw closing and the bubble collapsing is a sequence of events that pass through domains of physics, usually associated with very different scales of energy and mass. The water jet forms. The pressure differential opens a small vacuum in the water's body. The vacuum fills with vapor. The vapor bubble expands briefly before the surrounding pressure overwhelms it and drives it back inward. In the last nanoseconds of that inward compression, the trapped vapor reaches temperatures that under any other description would suggest an entirely different class of event. And then it is over. The bubble is gone. The water is water again. The shrimp repositions its claw, and the sediment settles. What the shrimp is doing, in a functional sense, is controlling the geometry of a collapse. The claw is shaped to produce a jet in a specific direction. The jet creates a bubble at a specific distance from the claw tip in a specific part of the water column. The bubble collapses at that location. The shock wave radiates outward from that point. If the shrimp has aimed correctly, that point is close enough to its target that the shock wave arrives with sufficient pressure to incapacitate it. The chain from claw angle to bubble position to shock wave delivery is a chain of physical causation that the shrimp has not, in any cognitive sense, reasoned through. It is a chain that evolution has encoded in the geometry of the claw and the behavior of the animal. The shrimp does not understand cavitation. It does not need to. The claw is built in such a way that cavitation is what the claw produces, and the shrimp has learned, in the sense that its nervous system has learned, across a very short behavioral circuit, to position that claw in the way that makes cavitation useful. The range at which the shock wave is effective is small. A few centimeters is the scale typically described, close enough that prey needs to be nearly within reach before the snap will reliably stun or kill it. This means the snap is not a ranged weapon in any expansive sense. It is a close quarters event. The shrimp still needs to maneuver, still needs to bring its claw near enough to whatever it is hunting to place the bubble at the right distance. The speed and violence of the event itself might suggest an animal that can afford to be imprecise, but the geometry does not support imprecision. The bubble must collapse at the right place. The claw must be angled correctly. Before the snap, there is a moment, usually brief, sometimes extended, in which the shrimp makes small adjustments, tilting the claw by a degree or two, repositioning its body incrementally along the substrate. The adjustment is quiet and unhurried. The snap, when it comes, is not. It is this contrast between the deliberateness of the positioning and the instantaneousness of the snap that gives the pistol shrimp's hunting behavior a quality that is easier to appreciate in slow motion footage than in real time. In real time, the shrimp moves, pauses, snaps, and something nearby goes still. In high speed footage, slowed by a factor of thousands, the movements that precede the snap become visible, and the bubble that follows it becomes visible, and the collapse of the bubble into its final shockwave becomes something that can be watched frame by frame. Each frame represents a slice of time too small to exist as an experience for any biological observer, but accumulated into a sequence, they produce a picture of an event that is, at its own timescale, coherent and precise and physically complete. The question of how long the shrimp has been doing this, how old the mechanism is, in evolutionary terms, does not have a clean answer. The family Alphidae is ancient, and the snapping claw in its various forms appears across the more than 1,100 described species in ways that suggest the basic mechanism predates the diversification of many of its current lineages. The particular geometry of the plunger and socket claw, as it exists in the most powerful snappers, is a specialized refinement of a more general alpheid claw structure, and the degree of specialization suggests a long history of selection for exactly the physical events the most powerful snappers now produce. How long ago a common ancestor first produced something recognizable as a cavitation bubble when its claw closed is not known, but the mechanism's current form is not new, and the seafloors of tropical coastlines have been crackling with the sound of these collapses for longer than most of the reef structures they now inhabit. The water temperature matters to the snap. Warmer water is less viscous, and the dynamics of bubble formation and collapse vary with the properties of the medium. The effects of temperature on the precise parameters of the snap, its peak pressure, the duration of the bubble, the temperature at collapse, are areas of ongoing study, and the full picture of how the snap performs across the range of water temperatures pistol shrimp inhabit is still being assembled. What is clear is that the mechanism works well enough across a wide range of tropical and subtropical conditions to have persisted without fundamental alteration across the span of time that produced the diversity of the current family. Evolution does not preserve mechanisms that consistently fail. What is perhaps most interesting about the snap as a physical phenomenon, as an object of study, as a fact in the world, is how well it hides at the scale of ordinary observation. A diver swimming above a reef flat where pistol shrimp are active will hear crackling. If there are many shrimp in a small area, the crackling may be nearly continuous, a kind of textured background noise rising from the sediment. Individual snaps within that background are not always distinguishable from each other. The animal responsible for each one is small, typically out of sight, often inside a burrow. Nothing visible marks the moment of a snap. Nothing visible marks its target or its effect. The event that produces the crackling, the jet, the bubble, the nanosecond temperature spike, the shock wave, the collapse, happens entirely below the threshold of what the diver can see in a fraction of a second that the diver's nervous system is not built to resolve. The instrumentation required to study the snap properly reflects this gap. High-speed cameras recording tens of thousands of frames per second, photomultiplier tubes sensitive to single photons, hydrophones calibrated to the kilohertz ranges where the acoustic signal is strongest, laser-based flow visualization to track the movement of the water jet, pressure-sensitive sensors positioned at measured distances from the snap to map the shock wave's outward propagation. None of these instruments are perceiving something that is not there. They are perceiving something that is there, but that the unaided senses of the researchers using them cannot access directly. The instruments extend perception into a part of the world where the pistol shrimp already lives and operates without any such assistance. That asymmetry is part of what makes the snap interesting as a biological fact. The shrimp has evolved to function in a physical regime that human observation cannot reach without specialized technology. The event that the shrimp produces routinely as a means of obtaining food and defending territory required decades of developing imaging and detection tools before it could be adequately characterized. The bubble that collapses beside the claw tip was being produced by pistol shrimp across tropical seafloors for millions of years before a high-speed camera was pointed at one. The temperature spike inside that bubble was reaching thousands of Kelvin long before anyone had the instruments to measure it. The flash of light was occurring, briefly in every snap, before a photomultiplier tube in a darkened laboratory in 2001 confirmed that it was there. The snap of a pistol shrimp is, in the end, a sequence of ordinary physics. Nothing about what happens violates the equations that describe how pressure and temperature and phase change behave in fluids. What makes it striking is not that the laws are different for the pistol shrimp, but that the animal operates them at a scale and speed that produces extreme outcomes from a mechanism small enough to fit inside a claw, smaller than a human fingernail. The sun's surface temperature, briefly and locally reproduced beside a claw made of chitin on a sandy seafloor in the course of hunting a small fish or a passing shrimp. The conditions are extreme by any measure. The setting is not. It is one of the more quietly unusual juxtapositions that the living world has produced, and it continues to be produced without interruption wherever pistol shrimp are active, which is across the tropical and subtropical oceans, nearly everywhere within reach of shallow water and a suitable patch of sediment. The snap that produces all of these events is not rare. It is not special in the sense of occurring seldom. It is common in the way that breathing is common, a biological process performed repeatedly, at all hours, by an enormous number of individual animals across a very large portion of the planet's coastal waters. Each individual snap lasts a fraction of a millisecond and covers a physical space no larger than a few cubic centimeters of water, multiplied across all the animals performing the same event, across all the shallow tropical seafloors that currently host them. The aggregate becomes something else entirely, a continuous acoustic presence in reef ecosystems, a persistent source of cavitation events in the water column, a biological system operating at a combined scale that the individual animal does not suggest. But before any of that scale comes into focus, there is first just the single animal on its patch of seafloor. The claw angled a fraction of a degree to the left of where it was a moment ago. The stillness that precedes the snap, the closing of two half-gram pieces of chitin at 100 kilometers per hour, and the bubble that follows the jet that the closure produces, and the collapse that follows the bubble, and the 700 microseconds that contain, in sequence, a temperature approaching the visible surface of the sun, and a flash of light too faint for any eye to register, and a shock wave sufficient to stop a fish in the water a few centimeters away. And then the seafloor, as it was before. The pistol shrimp moves forward. The claw is already cocked again, the plunger held back by the muscle that will release it at the next moment of need. The sand around it settles back to whatever arrangement the previous snap disturbed. The water above the seafloor is water again, having been for a fraction of a millisecond, not quite. In a darkened laboratory, a graph still exists, printed or saved in the records of the 2001 study that shows a small sharp peak in photon count at the moment of bubble collapse. That peak is the shrimp luminescence, the light produced by the temperature, produced by the compression, produced by the collapse, produced by the jet, produced by the claw. The graph is not a dramatic document. It is a scientific record. But what it records is a connection between a small snapping claw on a tropical seafloor and a number, 4,700 Kelvin, 8,000 Kelvin, that belongs to the physics of stars. The connection is real. The laboratory in which it was confirmed is quiet now. The tropical seafloor where it continues to occur is not. A pistol shrimp on a sandy flat in the shallows of a coral lagoon pauses in its movement, brings its snapping claw to a particular angle against the substrate, holds that position for a moment, and adjusts just slightly, just enough, before going still again. The deliberateness of that final adjustment is visible in the high-speed footage. The stillness that follows it is complete, and then the claw closes, and the water does what the water will do when the pressure drops fast enough to vaporize it, and the bubble forms and falls and is gone, and the shrimp moves forward into whatever space the shock wave has cleared, carrying its claw held slightly outward, pale and translucent at the joint, already in the process of cocking again. A pistol shrimp forages just outside its burrow in the pale sediment of a reef flat, moving with the careful, deliberate quality of an animal that knows exactly where it is and approximately how far it is willing to go. One long antenna trails behind it, and to the side, resting lightly against the flank of a small gobbyfish lying still at the burrow entrance above. The antenna barely moves. The gobby barely moves. Between them, through that single thread of contact, everything the shrimp needs to know about the world above the sand travels downward in a language neither animal could be said to have invented. That thread of contact, one antenna, one flank, two animals going about their separate business at the same address, is what the partnership between pistol shrimp and goby fish is made of at its simplest level. The shrimp cannot see well enough to detect a predator at a useful distance, and the goby, lying at the mouth of the burrow with its eyes elevated slightly above the sediment surface, what the goby sees, the shrimp is informed of, not through sound, not through the chemistry of the water, but through the precise muscular language of a tail pressed against a patch of water that then moves against an antenna that is already listening for exactly this. The shrimp and the goby have arrived, across whatever slow accumulation of evolutionary time produced this arrangement, at something very close to a shared nervous system for the purpose of knowing when to run. Running, in this case, means reversing into the burrow. The shrimp does it fast and without hesitation when the goby signals correctly, the whole body folding backward into the tunnel entrance with the practiced ease of an animal that has performed this retreat many hundreds of times and expects to perform it many hundreds more. The goby, depending on the severity of what it has seen, will either remain at the entrance or follow the shrimp down. Small movements of the tail mean something different from large ones. A rapid, high-amplitude flick carries a different weight of information than a slow, low-amplitude one. The amplitude corresponds, in a rough and practical way, to the size or urgency of the threat. A shadow passing overhead at a casual angle reads differently from a fish descending with the specific purposeful posture of something that has decided to commit to the dive. The goby, which has learned to read the difference from experience and from whatever the species already carries, conveys it accurately to the animal underground that cannot see anything at all. The word for this kind of relationship, in the technical language of ecology, is mutualism. Both animals benefit. The shrimp contributes its labor, digging, excavating, reinforcing, rearranging, and receives in exchange the continuous environmental monitoring of an animal with substantially better eyesight. The goby contributes its eyesight and receives in exchange a burrow it could not have built and does not have to maintain. Neither animal would do as well alone. The shrimp without the goby would forage more cautiously, emerge less often, expose itself more rarely to the open sediment where food is found. The goby without the shrimp would have no burrow to retreat to, no shelter from the dark of night or the passing of predators too large to outswim, no enclosed space to sleep in when the reef above goes quiet. Together they occupy a narrow strip of seafloor with a confidence that neither would possess separately. About 100 to 150 species of pistol shrimp in the genus Alpheus have been documented in this kind of partnership, paired with approximately 120 species of goldfish, making this one of the most thoroughly mapped symbioses in the ecology of coral reefs. The pairing is not random. Individual animals in the wild tend to be found in specific combinations, particular shrimp species with particular gomy species, and the relationship, once formed, is generally stable for the lifetimes of the individuals involved. A shrimp and a goby that have been sharing a burrow for some months are, in a measurable sense, calibrated to each other, their communication more fluid, their response times shorter, their behavior around each other more relaxed than it would be around a stranger of the same species. The partnership is not merely structural, it is individual, and it takes time to settle into itself. Laboratory work has added something to this picture that is worth sitting with for a moment. When a pistol shrimp is paired experimentally with a goby of an unfamiliar species, one it has not cohabited with before, the shrimp's response to the goby's alarm signals is measurably slower than it would be with a familiar partner. The two animals are beginning the work of learning each other. The goby produces a tail movement, and the shrimp, instead of reversing instantly, pauses for a fraction longer than it would with a known companion, as though weighing the signal against an uncertainty that will diminish over days and weeks of shared life. The conclusion the researchers drew is that the alarm signal system is not purely instinctive in the sense of being complete and automatic from the moment two animals meet. It requires, in some functional degree, that the animals learn each other's specific version of the shared language. Two different species that both participate in this symbiosis may be speaking, on first encounter, what amounts to slightly different dialects of the same signal set, and some time spent together is part of what brings those dialects into alignment. There is something quietly interesting in this, the idea of two animals in a burrow on a reef flat, neither of them doing anything that would look to a casual observer like learning, and yet both of them in the early stages of becoming something to each other that they could not have been on the first day. The shrimp keeps its antenna on the goby's flank. The goby holds itself still at the entrance. Each signal that passes between them and results in the right response from the right animal is, in a small way, a confirmation of the translation. Over weeks, the latency shortens, the pauses disappear. What was calibration becomes fluency, and the two animals move through their shared day with the ease of partners who have been long enough together that their separate purposes no longer require much deliberate negotiation. The burrow itself is worth considering as a structure, not only as a place. A typical pistol shrimp burrow is not a simple tube pressed into the sediment. It is a branching U-shaped tunnel extending roughly 20 to 30 centimeters into the seafloor, with a geometry that reflects the range of things the shrimp needs it to do. There is a sleeping chamber, a widening in the tunnel where the two animals can rest side by side during the hours of darkness. There is a food storage alcove, a small side chamber where the shrimp moves material brought back from its foraging on the surface. And there is a brood chamber, deeper and more enclosed, where the female shrimp carries her eggs attached to the small paddle-like limbs beneath her abdomen, tending them carefully through the weeks before they hatch. The burrow is not just a hiding place. It is a home in the fullest practical sense, a structure organized around the different needs of different moments in the animal's shared year. The shrimp builds and maintains this structure continuously and alone. The goby does not dig. It rests, watches, signals, and occasionally brings scraps of food back into the burrow. But the physical labor of construction is entirely the shrimp's work, carried out with the large snapping claw and with the smaller walking legs that move excavated sediment in small loads toward the entrance and deposit it just outside in a neat mound that grows and is periodically redistributed. Over the course of a burrow's occupied life, the shrimp will have moved a quantity of material that is large relative to its own body, rearranging the tunnel after storms disturb the sediment above, reinforcing passages that have partly collapsed, opening new chambers when conditions in the existing ones change. The burrow is always a work in progress, and the shrimp is always the one doing the work. At the surface, the evidence of this work is a small mound of displaced sediment at the entrance, excavated sand and rubble piled at the threshold between the burrow's interior and the open water. It is an ordinary-looking feature of a reef flat, easy to overlook among the texture of the seafloor, easy to step over without noticing what it signifies. But standing just out from that mound, in the lower few centimeters of the water column above a shallow tropical seafloor, this small heap of material represents the accumulated labor of an animal roughly the size of a human thumb building and maintaining the infrastructure of a shared domestic life in the sediment below. The mound is both entrance and evidence, the outer face of everything happening underground. During the daylight hours, the shrimp and the goby work the space just outside the burrow entrance together. The goby lies or hovers near the entrance, positioned to see in the directions that matter most, given the local topography of the reef. The shrimp forages within a few centimeters, moving out to collect material and retreating back toward the burrow entrance regularly, the antenna contact maintained more or less continuously, as long as the shrimp is close enough for the thread to hold. When the shrimp moves too far for the antenna to reach, the contact breaks, and for that interval, the shrimp is operating without the goby's watch, foraging in short, careful excursions into the open sediment, and returning before too much time passes. The antenna contact is not constant in any absolute sense. It is maintained as a default and reestablished as a priority after any interruption. Both animals behave as though they prefer to have it in place. Food for a pistol shrimp is collected rather than hunted at the surface level of the burrow entrance. Small pieces of organic material, detritus, fragments of algae, small invertebrates are gathered from the sediment nearby and carried back. The snapping claw, in this context, is a digging and manipulating tool as much as anything else, used to move rubble and sediment as the shrimp works around the burrow entrance. The smaller walking legs handle delicate material. The chemoreceptors on the shorter antennules read the chemical texture of the water and sediment continuously, identifying what is worth picking up and what is not. The shrimp is, in its foraging, a careful and methodical animal, moving incrementally, assessing constantly, returning to the burrow with what it finds. The goby watches all of this, its eyes mounted on slightly elevated positions on its head, track movement at a distance the shrimp cannot assess for itself. A predatory fish moving along the reef edge, still too far away to represent an immediate threat, will register with the goby long before it becomes the kind of presence that demands a response. The goby reads the trajectory, it reads the speed, it reads the posture. Fish that are hunting carry themselves differently from fish that are simply passing through, and the goby, which has spent its entire life reading this particular visual field, has had extensive opportunity to calibrate these distinctions. When the assessment results in a signal, the signal is graded to the assessment. A distant, slow-moving, apparently disinterested predator produces a different tail movement from a close, fast, committed one. The shrimp, underground or at the surface, reads the grade of the signal and adjusts its response accordingly. What this produces in practice is a division of the world into inside and outside the burrow, mediated by a continuous low-level assessment flowing from one animal to the other through the point of physical contact between them. The goby manages the assessment, the shrimp manages the burrow. Each is the specialist in its own domain. And in the narrow band of the burrow entrance, where the two domains meet, the antenna rests against the flank, and the information moves. There is something in this arrangement that has a quality of quietness about it, not the quietness of inactivity, but the quietness of a system that has settled into itself and now runs without friction. Two animals side by side at the entrance of a tunnel in the sand, each doing something the other cannot do. The whole arrangement held together by the weight of a single antenna resting against a body. The reef above them carries its ordinary traffic of fish and current and light. The burrow below carries its ordinary temperature and darkness, and the soft movement of eggs tended by the female in the deeper chamber. And at the threshold between these two worlds, the partnership holds its position without requiring anything more than the ordinary continuation of what both animals have always done. The pistol shrimp partnership with goby fish is, in terms of the number of species involved on both sides, one of the richer examples of mutualism in the ocean. Over 100 species of shrimp and over 100 species of goby participate in some version of this arrangement across the full extent of the reef systems of the tropical Indo-Pacific and beyond. The specific pairings tend to cluster by habitat and depth. Certain shrimp species are found most often with certain goby species, not because there is some exclusivity to the arrangement, but because the two animals tend to share preferred habitats and encounter each other most often in the places they both already favor. A sandy flat at a particular depth on a particular kind of reef will tend to have its own characteristic combination of shrimp and goby species, the two animals having arrived at their partnership through proximity as much as through active selection. In some cases, the pairings are observed to shift when one partner is lost. A goby whose shrimp has died may wait at the burrow entrance for some time before departing, apparently reluctant to give up the shelter it has been using, and the burrow itself will typically deteriorate without the shrimp's ongoing maintenance. A shrimp whose goby has disappeared may continue to use the burrow, but will forage more cautiously, making shorter excursions into the open sediment, pausing more frequently before leaving the safety of the entrance. The absence of the partner changes the behavior of the remaining animal in ways that suggest each is, in some functional sense, oriented around the presence of the other. Neither animal has been found to form the partnership on a temporary or purely transactional basis. What the data shows consistently is two animals that settle into each other over time and whose separate behavior is shaped by the relationship they have built. The female pistol shrimp carries her eggs on her pleopods, the small paddle-shaped limbs under her abdomen, and tends them actively through the weeks before they hatch. She fans them with gentle movements that keep oxygenated water moving across the clutch. She removes particles that settle on them. She adjusts her position in the brood chamber to keep the eggs in conditions that favor their development. The male, where present, may remain in the burrow or nearby, though the extent of his contribution to brood care varies by species and is not uniformly documented. What is consistent is that the female's behavior changes around the eggs. She becomes less inclined to leave the burrow, more attentive to disturbance, and the full structure of the burrow, with its brood chamber positioned at the deepest and most protected point of the tunnel system, reflects the significance of this phase of the animal's reproductive cycle. The eggs, when they hatch, release larvae into the water column, small, transparent, and helpless in the way of all marine larvae, drifting in the plankton layer with the current for some weeks before settling to the seafloor and beginning the process of finding a burrow site, a goby partner, and a stable home in the sediment. Most will not complete this journey. The ocean's larval stages carry high mortality as a general condition. The ones that do settle will begin again the slow, ordinary work of digging into the sediment of a reef flat somewhere, starting a burrow from nothing, and waiting to see whether a goby of the right kind will take an interest in the entrance they have opened. Within the genus Synalpheus, a different kind of social organization has developed that moves the pistol shrimp's communal life into territory that is, in terms of how biologists classify social behavior, quite unusual for a crustacean. Species in this genus form eusocial colonies, the same structural arrangement of society found in many ant and bee species, in which a single dominant breeding female leads a group of non-reproducing workers who collectively maintain the colony's shared home and defend it against intruders. In the insect world, this arrangement is so common that it is almost a default expectation. Among crustaceans, it is rare, and among marine crustaceans, the Synalpheus pistol shrimp are among the clearest examples it occurs at all. The shared home, in the case of Synalpheus colonies, is not a burrow in the sediment, but a sponge. Certain sponge species that grow on coral reefs contain within their bodies an extensive system of canals and chambers, the same canals the sponge uses to filter seawater for food, and these internal spaces are large enough, in some species, to house a colony of pistol shrimp in their interior. The shrimp live within the body of the sponge, using its canal system as the architecture of their colony, with the dominant female in the deepest and most protected part of the sponge's interior, and the workers distributed through the branching tunnels throughout. When an intruder approaches the sponge, another shrimp of a competing species, or some other small animal attempting to enter the canal system, the workers respond with coordinated snapping directed outward from within the sponge's interior. The snaps do not arrive one at a time from a single defending animal. They arrive in a burst from multiple workers positioned throughout the canal system, the sound and pressure of multiple snaps overlapping and amplifying each other in the confined space of the sponge's internal structure. The effect is a defense that is considerably more forceful than any individual worker could produce alone, achieved through the coordination of many animals acting together in response to the same signal. The workers have no reproductive stake in the outcome of this defense. They are non-breeding individuals, and the colony's reproductive output passes entirely through the dominant female. What they defend is not their own reproductive opportunity, but the colony itself, the sponge, the queen, the structure of the shared home. The evolutionary logic of this arrangement, across all the species where eusocial behavior has developed, involves the degree of genetic relatedness among colony members. Workers in a eusocial colony are, in most well documented cases, so closely related to the queen and to each other that contributing labor to the colony's reproductive success is, in terms of genetic propagation, a more effective strategy than reproducing independently. The pistol shrimp workers in a Synalpheus colony are typically highly related to the queen. They are in most cases her siblings or offspring from earlier clutches, and the high relatedness that characterizes these groups is consistent with the evolutionary conditions that, in the theoretical framework developed to understand eusociality in general, tend to produce it. Whether the shrimp experience anything like the social cohesion that the structure implies is a question the data cannot answer. What the data shows is the structure itself, the queen, the workers, the coordinated defense, the shared sponge, and the tight genetic relatedness, tying all of it together. Colony sizes in Synalpheus vary considerably by species, ranging from small groups of fewer than 10 individuals to larger colonies of several hundred. In smaller colonies, the distinction between the queen and the workers may be subtle in terms of body size. In larger ones, the queen is often noticeably larger than the workers, her body shaped in part by the sustained reproductive activity that defines her role in the colony's structure. The workers remain smaller, their reproductive organs undeveloped or minimally developed, their bodies oriented toward the labor of defense and maintenance rather than toward the production of eggs and larvae. The sponge that houses the colony is itself a living animal, and the relationship between the shrimp and the sponge sits somewhere between inhabitation and mutualism. The shrimp benefit from the sponge's architecture, and there is some evidence that the sponge benefits from the shrimp defensive activity, which may deter other organisms from damaging or competing with the sponge's tissue. The degree to which this constitutes a true mutualism of the kind that characterizes the shrimp-goby partnership is not fully established. What is clear is that specific Synalpheus species are consistently found in specific sponge species, and the association is not random. The shrimp have preferences, and the preferences are precise. A Synalpheus colony encountered in the field presents a particular kind of quietness. From the outside, the sponge looks like a sponge, a rounded, irregular mass of slightly rubbery tissue attached to the reef substrate, its surface perforated with the small openings through which it draws and expels water. Nothing about the exterior announces that the interior is occupied. The first indication that something lives inside comes in most cases from sound, a crackle or pop from within the sponge's body when a hydrophone or listening device is brought near, the workers snapping in the interior tunnels, the sound carrying outward through the sponge's tissue and the water around it. To a diver passing the sponge without specialized equipment, the sound might not register at all. The colony is hidden inside its home in a way that the shrimp-goby partnership at an open burrow entrance is not, and the privacy of its domestic life is accordingly more complete. The dominant breeding female in a Synalpheus colony is, in a sense, the living center of the colony's future, the only animal through whom the colony's genes pass into the next generation, the only one whose reproductive output accumulates into the population of workers that keeps the whole structure running. When she dies or is experimentally removed, colonies show measurable behavioral change. Workers in colonies that have lost their queen become more active at the outer edges of the sponge's canal system, and in some cases, a new female eventually assumes the reproductive role, though the process of transition is not uniform across species, and the details vary. The colony does not simply dissolve when the queen is gone. It reorganizes, slowly and imperfectly, around whatever new arrangement the remaining animals can construct from the materials the colony already has. The workers, going about their day inside the sponge's canals, are not waiting for a crisis to give their activity meaning. They move through the tunnel system of the sponge's body in the ordinary course of the colony's daily life, grooming each other, tending eggs in the queen's chamber, clearing material from the canals, positioning themselves at the sponge's surface, openings in the posture of animals prepared to snap if something approaches too closely. The coordination among them is not managed by any visible central authority. There is no worker who directs the others, no foreman position, no hierarchy among the non-breeding members. The coordinated defense emerges from the aggregate response of many animals, each responding, in parallel, to the same environmental signal. The result looks organized from the outside. The mechanism, as far as researchers can determine, is distributed through the colony rather than centralized within it. This distributed quality is present in the shrimp-goby partnership as well, in its own way, two animals, each attending to the part of the shared situation that falls within its competence, producing together a result that neither could achieve alone, without any directing authority between them. The shrimp does not tell the goby where to look. The goby does not tell the shrimp when to dig. Both animals pursue their separate purposes within the shared structure of the burrow and the partnership, and the outcome of their separate pursuits is a home in the sand that works reliably and continuously for both of them. In the quieter hours of the afternoon, when the reef above the burrow holds the particular quality of filtered light that characterizes a tropical lagoon in the middle of the day, the light diffuse and warm, moving slightly with the surface current far above, the shrimp and the goby slow down. The shrimp's foraging excursions become shorter and less frequent. The goby's posture at the entrance relaxes incrementally, its body resting more fully against the sediment, its eyes still open and still attending to the middle distance, but with the slightly softened attention of an animal that has decided, provisionally, that the afternoon presents no immediate concern. The antenna may lose its contact with the goby's flank for longer intervals. The burrow entrance sits undisturbed. The small mound of excavated sediment at the threshold holds its shape in the light. As the afternoon light begins to shift, the color temperature changing, the angle of the filtered rays steepening toward the slower illumination of the late afternoon on a shallow reef, the two animals begin the transition toward evening. The shrimp moves less. The goby, whose body language the shrimp has spent months learning to read, takes on the particular stillness that precedes its descent into the burrow for the night. The goby pauses at the entrance longer than it has paused at other moments during the day, a moment of attending to the water above with the full alertness of an animal performing one last assessment before withdrawing. Then it lowers itself into the burrow entrance, moving backward and down into the tunnel with the smooth, practiced ease of a body that knows every centimeter of the space it is entering. Below, in the sleeping chamber, the shrimp has already drawn itself into the quiet posture of an animal at rest. The chamber is dark and still. The temperature of the sediment slightly more stable than the open water above, the current that moves across the reef flat, barely perceptible at this depth. The goby settles beside the shrimp in the chamber. The antenna, no longer needed for the transmission of alarm, rests. The eggs in the brood chamber deeper in the burrow continue to be tended in the ordinary ongoing way of eggs being carried, the female's pleopods moving in slow periodic strokes, the eggs receiving the oxygenated water that keeps them viable. The work of the burrow does not fully stop when the animals sleep, because some of what the burrow needs is continuous and does not wait for morning. The reef above the burrow goes on in its nighttime way, the diurnal fish retreating, the nocturnal ones emerging, the current continuing across the flat in its slow tide-governed movement, the water carrying its load of planktonic organisms through the darkness above the sediment. None of this reaches the sleeping chamber in any form that would disturb what is resting there. The shrimp and the goby lie in the enclosed quiet of the space the shrimp has built and maintained through the weeks and months of their shared tenancy. The darkness of the burrow interior complete, the warmth of the sediment around them steady and even. Outside, at the burrow entrance, the small mound of excavated sediment sits in the dark water at the threshold between the interior of the burrow and the open reef flat above. It is the accumulated labor of a small animal going about the ordinary work of its ordinary life in the sediment of a shallow tropical lagoon. The night passes over it. The current moves lightly across its surface. The mound holds its position at the lip of the burrow entrance. In your sport brekst work steering until next special by the pastil one. The outer edge of the world the shrimp has made, sitting quietly in the dark water of a reef flat, somewhere above the equator, while the two animals below sleep side by side in the chamber. They have come, over weeks of shared signals and calibrated attention, to consider home. A hydrophone suspended above a shallow reef in the Indo-Pacific picks up something that is not quite a sound and not quite a texture, a continuous, overlapping crackling that rises from the seafloor below and fills the water column from the sediment to the surface, present at every hour, neither louder nor quieter at noon than at midnight, neither pausing nor building, but simply persisting, a sound as ambient and constant as the reef itself. It is not a single animal. It is not a single event. It is the product of millions of small claws opening and closing in the dark of the sediment, each snap discreet and brief, but arriving so frequently and from so many directions at once that what reaches the hydrophone is not a sequence of pops, but something closer to an unbroken surface of crackling static, a texture the ear reads as continuous, even though, if the recording were slowed to a small enough window of time, it would resolve into thousands of individual events, separated by tiny intervals of silence. This is the sound of a living reef in its resting state. Acousticians who study shallow tropical waters use a phrase for what the hydrophone records in places like this: snapping shrimp noise. The description is plain and functional and does not quite prepare a listener for the reality of it. The crackling produced by dense pistol shrimp populations dominates the frequency band between roughly 2 and 15 kilohertz across shallow reef environments worldwide, a band that covers much of the range most useful for listening, the range in which a great deal of underwater sound travels most clearly and most far. Within that band, in the places where the shrimp are most dense, the snapping is not a background element competing with other sounds. It is the acoustic floor. Everything else the reef produces, the low thrumming of passing fish, the clicks of other crustaceans, the distant percussion of waves on coral, sits on top of it, the way birdsong sits on top of wind. The quality of the sound has been compared, in scientific literature and in the field notes of researchers who have listened to hydrophone recordings for long hours, to the crackling of frying bacon. Others describe it as static, or as the sound of rain on dry leaves heard from inside a room, or as the particular texture of interference on an old radio tuned between stations. None of these descriptions are quite right, because each of them implies something sporadic or irregular, something that rises and falls with a source that changes over time. What the hydrophone records from a dense shrimp population does not rise and fall, it simply continues. The crackling has been described as the loudest biological sound source in shallow tropical waters, louder in its frequency band than passing ships, louder than rain on the surface, louder than the collective movement of fish schools that may contain tens of thousands of individuals. It is produced by animals no larger than a human thumb, each one acting entirely alone, generating a sound no louder than a single sharp pop, and together, in the millions, generating something that fills an ocean. The reef flat of a healthy atoll in the tropical Pacific on a clear day with little wind is one of the quieter places in the human sensory world above the waterline. The water is shallow and warm and almost motionless. The surface moves in long, slow swells. The sand below is pale and still. There is very little to hear from above. Below the surface, the hydrophone records something else entirely, the uninterrupted crackling of a population that does not rest, spread across the sediment from the reef crest to the sandy lagoon floor, each individual shrimp going about its own particular work of foraging and defending and maintaining, each snap a private act that joins with every other private act to produce, collectively, the dominant sound of this place at every hour of the day and night. The density of snapping shrimp populations in healthy reef environments can reach tens of thousands of individuals per hectare, concentrated in the rubble zones and sandy patches and coral margins where the burrows are easiest to excavate and the food supply most reliable. At those densities, the overlap between individual snapping events becomes nearly complete. There is almost never a moment across a large enough section of reef when no snap is occurring. The crackling is not a storm that passes through the reef at intervals. It is the reef's continuous, unremarkable acoustic background, the sound it makes while nothing in particular is happening. In the early 1940s, this was not common knowledge. It was not even uncommon knowledge. It was simply not known. Allied naval forces operating in the Pacific during the Second World War encountered, in shallow coastal and reef waters, a persistent acoustic interference that resisted every attempt at explanation. Passive sonar systems, which worked by listening for the characteristic sounds of enemy submarine engines and propellers and hull vibrations, depended on being able to isolate those sounds from the ambient noise of the surrounding water. In deep open ocean water, the ambient noise was manageable. It had known sources and known behavior, but in shallow coastal waters near reefs and in the protected bays and inlets used as transit routes, the passive sonar systems picked up something that had no obvious source at all, a continuous crackling, dense and persistent, and spread across exactly the frequency ranges most useful for detecting submarines, producing interference so complete that the acoustic signature of a submarine could not be reliably separated from it. The interference was consistent enough and widespread enough that it became a recognized problem rather than an occasional anomaly. Operators reported it in sectors across the Pacific, in waters near coral reefs and seagrass beds, and sandy coastal shallows. It was present regardless of weather, regardless of surface conditions, regardless of time of day. It did not behave like interference produced by a mechanical source, which would have a direction and a pattern. It did not behave like the acoustic signature of a geological event, which would be transient. It simply persisted, filling the headphones of sonar operators with a crackling that was louder than what they were trying to hear, and came from everywhere at once. The explanation, when it came, was not arrived at quickly. Researcher Gordon Hamilton, working in the 1940s, produced detailed analyses of the acoustic contributions of snapping shrimp to shallow water ambient noise, establishing that the crackling interference the naval sonar systems were encountering was produced not by machinery or weather or geological activity, but by the accumulated snapping of dense populations of small crustaceans living in the sediment and rubble of the shallow seafloor. Hamilton's work helped to establish that the biological inhabitants of the ocean were not acoustically passive, that they were in fact major producers of the ocean's sound environment, contributing noise at levels that mattered for any system that depended on listening to the water. The analyses he published in this period are still referenced in modern ocean acoustics literature. They help to formalize, among other things, the discipline of bioacoustics, the study of sound produced by living organisms and its effects on the acoustic environment, as a field with recognized scientific and practical importance. The realization that small crustaceans were responsible for a significant portion of the ocean's acoustic background was at the time a recalibration of assumptions about which inhabitants of the sea were worth attending to when the question being asked was about sound. The large, the visible, the dramatic were easier to account for. Fish schools produced detectable noise. Whales produced unmistakable sound. Waves and wind and the movement of sediment were understood as acoustic contributors. The snapping shrimp were not large, not visible from the surface, not individually dramatic. They were present in numbers that made them collectively the dominant acoustic contributor in their frequency range, and that fact had gone unexamined until the acoustic signature they produced became an operational problem that demanded explanation. Hamilton's foundational models described the statistical behavior of snapping shrimp populations as acoustic sources, the way the distribution of snapping rates across a population produced the characteristic spectral shape of the interfering, the way depth and temperature and habitat type affected the overall level of the crackling, the way the overlap of individual snapping events at high population densities produced a sound that was in practice continuous. Those models set the terms for how researchers understood shallow water ambient noise for decades afterward, and they established snapping shrimp as a baseline acoustic reference in a field that would expand considerably as the tools for listening to the ocean improved. What a hydrophone records from a reef in the present is essentially what it would have recorded from the same reef in 1942 or in 1892 or at any point across the long history of the reefs themselves. The crackling was not produced by the war. It was not new. The ships and the sonar operators had simply arrived in waters that had been producing this sound continuously for far longer than there had been anyone to listen for it, and they had encountered it the way any new observer encounters a process that has been running without interruption, as something that appeared to require explanation, but had, in fact, simply been going on. Contemporary researchers studying the relationship between snapping shrimp and reef acoustics have access to instruments that were unavailable to Hamilton and his contemporaries, long-term autonomous hydrophone deployments that can record continuously for months at a time, logging every acoustic event across a wide frequency range without the need for a human observer to be present. These deployments have made it possible to study not only what the crackling sounds like at a given moment, but how it changes across time and in response to changing environmental conditions. One finding that has emerged from long-term acoustic monitoring is that snapping rates vary with water temperature in a consistent and measurable way. Shrimp in warmer water snap more frequently than shrimp in cooler water. The relationship is not dramatic at the scale of ordinary seasonal variation, but it is reliable enough to be detectable in hydrophone records, and it has implications for how the acoustic environment of coral reef ecosystems may change as ocean water temperatures rise over time. Warmer water means more frequent snapping, which means a louder and more acoustically dense environment, a shift not large enough to register as a sudden change in any single recording, but visible in the data when records from different temperature regimes are compared across months and years. The relationship between temperature and snapping rate is in itself a small fact. A warmer shrimp snaps more. This is not surprising in the context of the broader biology of ectothermic animals, whose metabolic rates and behavioral rates increase with temperature in a general pattern that holds across many species. What makes the relationship worth noting in the context of reef acoustics is that snapping shrimp are sufficiently numerous, sufficiently widespread, one acoustically sufficiently dominant in their frequency range, that changes in their snapping behavior produce changes in the acoustic character of the reef that are large enough to measure. The reef sound is partly a function of its temperature. The crackling that fills a hydrophone recording on a warm day is in a small but real way different from the crackling on a cool day, denser, faster in its texture, slightly higher in overall level. The reef is listening to itself change, in the sense that its own acoustic output records the conditions in which its inhabitants are living. Conservation researchers have taken this a step further, examining what the acoustic profile of a reef can reveal about its ecological condition. A healthy reef section and a degraded reef section, one that has experienced coral bleaching or chronic disturbance or a reduction in the complexity of its physical structure, differ in ways that are visible to a diver but also audible to a hydrophone. The degraded section is quieter, not silent, but measurably less acoustically dense than the healthy section adjacent to it. The crackling thinner and less continuous. The texture of the recording different in a way that the data can characterize precisely, even before any visual survey has been conducted. The reason for this is partly direct and partly indirect. The direct reason is that degraded reef sections support lower densities of snapping shrimp, because the complexity of the physical habitat that makes dense burrow construction possible has been reduced, and because the food resources that support large populations are less available. Fewer shrimp means fewer snaps means a quieter recording. The indirect reason is that snapping shrimp abundance is itself an indicator of broader reef condition. The shrimp are not isolated actors, but participants in an ecosystem, when the same processes that degrade the reef tend to reduce the abundance of the animals that produce its acoustic signature. This has led to proposals for using acoustic monitoring as a tool for assessing reef health, deploying hydrophones in reef systems, and using the acoustic complexity of the snapping shrimp signal as a bioindicator, a measurable proxy for the ecological condition of the reef. The hydrophone is listening to. The appeal of the approach is practical. Hydrophones can be deployed in places difficult for divers to reach. They can record continuously without the intermittent sampling limitations of visual surveys, and they can detect changes in acoustic profile before those changes would be obvious from visual inspection alone. A reef beginning to decline would, on this model, produce a detectable shift in its acoustic record before the visual evidence of that decline had fully manifested on the reef surface. The comparison, when researchers set a degraded reef recording beside a healthy one and examine both, is audible as well as statistical. The healthy recording has a density to it, a fullness, a texture that is almost tactile in the way it fills the frequency range, while the degraded recording has gaps in it. Not silences exactly, but a thinness in the texture, a reduction in the overlap between individual snapping events that makes the discrete pops more individually audible and the continuous crackling less complete. The two recordings sound different to a trained listener, and they look different on a spectrogram and the difference between them carries information about the condition of the place each one was made. A healthy reef, heard through a hydrophone, sounds busy in a particular way. Not chaotic, not distressed, but occupied, layered with the sounds of many animals going about their individual existences in the same water column at the same time, the snapping shrimp underlying everything else in their frequency range, the other sounds of the reef sitting above and around that continuous crackling like the components of a piece of music that has been playing for so long that no one can remember when it started. A degraded reef, heard through the same instrument, sounds different in a way that is difficult to characterize as anything other than reduced. Less of everything. The crackling is still present because the shrimp are still present in smaller numbers, and because the process of degradation is rarely complete. But the texture has changed. The frequency band is less full. The overlap between events is less complete. The sound of the place has become thinner in proportion to the thinning of the life that produces it. What the hydrophone records, in both cases, is not the reef's condition directly, but the acoustic output of the animals that live in it, and those animals, in their accumulated behavior across millions of individual acts, produce a record of the reef that is continuous, detailed, and available to anyone with an instrument capable of listening to it. The crackling is not a signal the reef is sending. The shrimp are not communicating with researchers or reporting their own status. They are simply going about the particular work of their existence, snapping and foraging and maintaining their burrows in the sediment, and the collective acoustic product of all that private activity turns out, when read carefully and over time, to carry information about the place where it is happening. There is something that takes a moment to absorb in this. The idea that an animal spending its life in a small tunnel in the sediment, attending to the local business of its own survival, contributes to a signal that is large enough to interfere with naval sonar and sensitive enough to indicate the health of an entire ecosystem. The shrimp is not doing any of this deliberately. It is not oriented toward any of these outcomes. It is managing its claw and its burrow and its partnership with the goby that rests at the entrance above, and the snap it produces to handle whatever requires handling in its immediate vicinity, joins, without any intention on its part, with the snaps of every other shrimp on the reef, and the sum of those joins becomes the acoustic signature of the reef itself. The crackling of a healthy reef in the Indo-Pacific, recorded on an autonomous hydrophone at 2 in the morning, with no ships nearby, and no weather on the surface, and nothing in particular happening across the dark expanse of the lagoon, is almost entirely the product of animals that cannot hear it, as a collective sound. Each shrimp hears, if it hears at all in the sense that the word usually implies, what is happening immediately around it, the snap of its own claw, perhaps the snapping of a close neighbor, the movements in the water that its antennae register as vibration and pressure, the large sound, the unbroken crackling that fills the hydrophone recording and covers the frequency range most useful for submarine detection, is not experienced by anything that produces it. It is a property of the population as a whole, an acoustic phenomenon that emerges from the aggregation of millions of individual acts and exists only at a scale none of those individuals occupies. This is a feature of many large biological sounds. The dawn chorus of birds in a temperate woodland, heard from a distance, is an acoustic event that no individual bird is attempting to produce. Each bird sings for its own reasons, in its own immediate context, without knowledge of or orientation toward the large sound that the chorus collectively generates. The crackling of a shrimp reef operates on the same principle, at a greater scale of production and a lesser scale of individual behavioral sophistication, and produces something that has proven ecologically significant and historically consequential in ways that have nothing to do with the intentions or experiences of any of the animals involved. Gordon Hamilton, listening to early hydrophone recordings in the 1940s and working to account for the interference that was troubling Allied sonar operators, would have been attending to something that the shrimp had been producing continuously for millions of years before anyone thought to listen for it. The reefs were old, the shrimp were old, the crackling was old, the sonar systems were new, and the novelty of the sonar systems was what made the old crackling suddenly visible as a phenomenon requiring a name and an explanation. This pattern, a biological process persisting for a very long time and becoming legible as a scientific fact only when a new instrument or a new practical problem creates the conditions for noticing it, is common in the history of natural science. And the pistol shrimp's contribution to ocean acoustics is one of the cleaner examples of it. The shrimp had not changed, the reef had not changed, the sound had not changed. What changed was the presence of a technology sensitive enough to be affected by the sound, and the presence of people who needed to understand what was affecting it. The autonomous hydrophone that records the nighttime crackling of a healthy Indo-Pacific reef today is a descendant, in a lineage of instrument development spanning 8 decades, of the listening systems whose interference prompted Hamilton's analyses. It is more sensitive, more stable, more capable of continuous unattended deployment. It logs data not to support naval operations, but to track ecological change across months and years, building records long enough to show trends that a single survey could not reveal. The sound it records is the same sound the wartime sonar operators heard in their headphones, produced by animals doing the same things in the same kinds of places, generating the same crackling that has been rising from reef sediment for as long as there have been pistol shrimp living in it. At the edge of a seagrass bed in shallow tropical water, where the sand gives way to the dense leaf canopy of the grass, and the light from above filters down in moving patches, the acoustic environment is its own version of the reef crackling, slightly different in character, shaped by the different density and distribution of shrimp in a habitat with its own particular structure. The seagrass shrimp snap at their own rate, maintaining their own burrows in the sandy substrate beneath the grass blades, and the sound they produce collectively is part of the acoustic identity of this habitat, just as the reef crackling is part of the reef's fall. Different habitats, different densities, different acoustic textures, but the same process, the same animal, the same fundamental mechanism, producing the same basic sound across the full range of the environments where pistol shrimp have found their way to live. The crackling follows the distribution of the shrimp, which follows the distribution of habitable coastal seafloor across the tropical and subtropical world. Where there are reefs, it is loudest. Where there are seagrass beds, it continues at a different character. Where there are mudflats and sandy shallows, and the complex rubble zones where reef meets sand, it persists in whatever density the habitat and the population support. There is, across a broad band of coastal tropical waters in the Pacific and Indian and Atlantic Oceans, effectively no location on the shallow seafloor where pistol shrimp are absent entirely, and so there is no location where the crackling is entirely absent. It is a persistent feature of this zone of the ocean, one of its defining acoustic characteristics, part of what shallow tropical water sounds like, as opposed to deep oceanic water or cold water systems where the shrimp do not live. The sound has been present, essentially unchanged in its character, since before any human civilization developed the capacity to listen for it. It was present when the first Polynesian navigators crossed the Pacific, their hulls passing above reefs whose acoustic signature none of them had reason to seek out. It was present when the first explorers dove on coral reefs and came up describing a silent world, which is what the world beneath the surface seems like to unassisted human hearing, because the frequencies at which the shrimp are most active do not carry well through the air-water interface and do not register clearly to ears evolved for hearing in air. The reef's silence was always an artifact of the instruments available for listening to it. When the instruments improved, when hydrophones became sensitive and portable and deployable, when recording equipment could capture and preserve what the water carried, when the data could be analyzed and compared and set against ecological surveys of the same sites, the crackling became audible in a new way, not just as interference, but as information. The same sound that had disrupted wartime sonar turned out, when the right questions were asked of it, to be a record of the reef's condition. The animals producing it had not changed their behavior to accommodate this new role. They had simply continued, as they had always continued, snapping in the dark of the seafloor, each one attending to its immediate world. An autonomous recording unit drifts very slowly above a tropical reef at night, suspended by a float at a depth of a few meters, its hydrophone pointed downward toward the seafloor below. The surface above is dark and still. There are no ships within range. The reef is in the middle of the hours between midnight and dawn, the quietest hours for human traffic in these waters, and the recording the instrument makes during this interval will show, when it is retrieved and analyzed, the same uninterrupted crackling it has shown during every interval of this deployment, the same texture, the same density, the same continuous acoustic surface generated by the same animals going about the same activity in the same sediment below. The water above the reef moves in long, slow oscillations, the faint remnant of swells that cross thousands of kilometers of open ocean before arriving here and diminishing to almost nothing in the shallow lagoon. The moonlight, where it reaches through the water, traces moving patterns across the pale sand and the rubble and the coral structures rising from the floor. The pistol shrimp, in their burrows and at their burrow entrances, and in the narrow zones of sediment, they work in the dark, snap without reference to the moonlight, or the recording instrument, or the long quiet history of the sound they are making. The crackling continues through the remainder of the night and into the pale early light of the following morning, neither louder nor quieter than it was at midnight, neither faster nor slower, the same texture it has been since before any instrument existed to record it, produced by animals that have no awareness of the scale of the thing they collectively make, only the small immediate fact of the claw, the burrow, the water, the dark. A mantis shrimp's dactyl club makes contact with the shell of a snail on a reef substrate, and the shell does not crack so much as it ceases to be intact, the two states separated by an interval too brief to have a useful name. The club has traveled a distance of a few centimeters in approximately 3 milliseconds, accelerating at roughly 10,000 times the force of gravity and arriving with a force of up to 1,500 newtons, which is enough to fracture the wall of a glass aquarium if the shrimp is positioned close enough to it and inclined to try. For a moment after the strike, in the water immediately surrounding the tip of the club, a small cavitation bubble forms and collapses, the same physical process, arrived at by a completely different route, through a completely different body plan, on a completely different branch of the crustacean family tree. The mantis shrimp did not inherit this from the pistol shrimp, and the pistol shrimp did not inherit it from the mantis shrimp. Each arrived there separately, working with what it had across millions of years of gradual modification, and the result in both cases was a creature that produces as a byproduct of its ordinary life one of the more extreme physical events occurring on a coral reef on any given afternoon. The mantis shrimp belongs to the order Stomatopoda, a lineage that has been distinct for somewhere in the region of 400 million years, which places its origins before the first forests appeared on land. There are roughly 550 known species, ranging in length from a few centimeters to around 30, and their coloration spans a range that includes some of the most vivid pigmentation found in any marine animal: deep blues, oranges, greens, and reds, arranged in patterns that in some species seem to serve no obvious camouflage function and may instead be involved in communication between individuals. The eyes of a mantis shrimp are among the most structurally complex visual organs in the animal kingdom. Each eye moves independently on a stalk, and the visual system contains up to 16 types of photoreceptor cells, compared to 3 in a typical human eye, giving the shrimp sensitivity across a range of wavelengths that extends well into the ultraviolet and the near infrared. What the mantis shrimp actually experiences as it looks out at the reef around it, what the world looks like from inside that visual apparatus, is genuinely unknown and probably unknowable in any meaningful sense. The dactyl clubs themselves are built to withstand what they deliver. High-speed photography has shown that the club strikes with enough force to generate both the direct impact and a second impact from the collapsing cavitation bubble that forms immediately afterward, so the target receives, in rapid sequence, 2 distinct strikes from a single swing. The club's outer surface is a helicoidal fiber composite, a structure in which mineralized fibers spiral around the impact region in a pattern that distributes and absorbs the stress of the strike rather than concentrating it at the surface. Materials scientists studying the structure have described it as one of the more effective natural impact-resistant architectures yet examined, and there is ongoing research into how similar arrangements of layered, rotating fiber composites might be used in engineering contexts. The shrimp rebuilds this surface periodically as part of its molting cycle, growing a new club each time it sheds its exoskeleton, a process that leaves it temporarily vulnerable in a way that a creature capable of shattering aquarium glass might find uncomfortable. Though what the shrimp finds is not something the research can tell us, there are 2 broad categories of mantis shrimp, loosely divided by how they catch things. Smashers use the dactyl club in the way that has attracted most of the attention: the high-speed impact strike against hard-shelled prey like snails, crabs, and clams. Spears use a different approach, extending long barbed appendages in a rapid outward thrust to catch soft-bodied prey like fish and shrimp before they can move away. The 2 groups have different habitat preferences and different temperamental profiles, at least in the sense that smashers in captivity have a documented tendency to attempt to break their enclosures, while spearers are more commonly described as remaining still and waiting. Both types are territorial, both maintain burrows, both have been observed producing signals, low-frequency sounds, fluorescent markings, behavioral displays that appear to carry social information, though the full repertoire of communication in any mantis shrimp species is not well mapped. The reef is full of conversations in channels that were not suspected to exist until someone thought to look. Electrophorus volti lives in an entirely different world, the murky, slow-moving rivers and flooded forests of South America, far from any reef, in water where light penetrates only a short distance, and the usual visual channels of the aquatic world are reduced to dim suggestions of shapes moving through brown water. This species, formally described as distinct from its relatives only in 2019, holds the measured record for the highest bioelectric discharge of any known living animal, up to 860 volts, produced by 3 pairs of specialized electric organs that together account for approximately 80% of the animal's total body volume. It is a long cylindrical animal, somewhat eel-shaped though not closely related to true eels, and the overwhelming majority of its body is in terms of internal structure, a living electrical generator. The muscles that most other elongated fish rely on for their length are in electrophorus volti, largely replaced or supplemented by stacked columns of modified muscle cells called electrocytes, each producing a small voltage increment, the increments adding together down the length of the body in the way that batteries in series add together to produce a cumulative charge. The discovery that there were multiple distinct species within what had long been treated as a single taxon came gradually through a combination of genetic analysis and closer physical measurement of specimens from different river systems across the Amazon and Orinoco basins. The voltage record of 860 volts was measured from a specimen of Electrophorus volti collected in the Erie River. Earlier measurements taken from animals that were likely a mix of the three species now recognized had produced figures around 600 volts, which had itself long been considered extraordinary. The additional voltage found in Electrophorus volti is thought to result from its greater body length, since the electrocytes are arranged in series and a longer body can accommodate more cells in sequence. There is something slightly difficult to hold in mind about an animal that is 80% electrical organ, that the shape moving slowly through the dark river water is mostly structurally a battery with fins. The discharge is used for navigation, for communication with other individuals of the species, and for prey detection. Electric eels produce weak, continuous electric fields and sense distortions in those fields caused by nearby objects, giving them a detailed picture of their immediate environment that does not depend on light at all. The high voltage discharge is used less continuously and more selectively. Research by Kenneth Catania at Vanderbilt University, published across several studies in the 2010 S, documented some of the more precise and counterintuitive aspects of how the discharge is deployed in practice. When Electrophorus encounters prey that is hidden or submerged in leaf litter, it can produce a rapid doublet or triplet of high-voltage pulses that cause involuntary muscle contractions in the hidden prey animal, causing it to move, revealing its position. The hunting strategy involves, in other words, using the electric discharge not to kill or stun initially, but to locate, by provoking an involuntary signal from the thing being searched for. The prey reveals itself. Then the eel can find it and direct a more sustained discharge at close range. What the dark river contains on an ordinary night is an animal moving slowly through water it cannot see clearly, reading the electrical texture of the space around it, occasionally sending out a pulse that causes something hidden in the mud to flinch, and then moving toward the flinch. There is no drama in this from the eel's perspective, no drama that can be confirmed at least. It is simply how the animal finds things, in the dark, in the water, in the particular river system where it has always lived. The bombardier beetle moves through leaf litter and soil in a way that does not particularly announce what it is carrying. It is a ground beetle in the family Caribidae, a large and diverse group with thousands of species, and from the outside it resembles other ground beetles closely enough that there is nothing in its ordinary appearance to suggest it contains, in its abdomen, a chamber capable of producing a scalding pulsed chemical spray directed with considerable accuracy across a sweep of nearly 270 degrees. The mechanism is a two-chamber system. One compartment holds a mixture of hydroquinone and hydrogen peroxide. The other holds a mixture of peroxidase and catalase enzymes. When the beetle is threatened, the contents of the two chambers are forced together into a reaction chamber where the enzyme mixture causes a rapid exothermic decomposition, a chemical reaction that generates heat and produces oxygen gas simultaneously. The oxygen creates pressure. The pressure forces the hot liquid out through a flexible nozzle at the tip of the abdomen in pulses at temperatures that reach approximately 100 degrees Celsius and at a rate of up to 500 pulses per second. The nozzle is steerable. The beetle can direct the spray forward, backward, sideways, and at a range of angles in between, a capability that has been demonstrated in laboratory settings by provoking the beetle from different directions and filming the response. When a spider approaches from the front, the nozzle angles forward. When an ant grips one of the beetle's legs, the spray is directed toward the leg in question. The accuracy is not precise in the way that a targeted instrument is precise, but the range of motion and the speed of response are sufficient to cover most angles from which a threat might arrive. A well-documented case, reported from field observations and later tested experimentally, involves a beetle that is swallowed by a toad or frog and discharges inside the predator's mouth and throat. The frog's swallowing reflex reverses. The beetle is expelled, typically alive, onto the leaf litter or ground. Several such beetles retrieved after this process were observed to groom themselves and walk away. The frog in the accounts also appeared to survive, which gives the encounter a quality that is more startling than grim. Two animals involved in a moment of genuine mutual inconvenience, each recovering and continuing. The chemistry that makes this possible is not unique to bombardier beetles. The two chemical precursors are found in many organisms, and the enzymes that catalyze the reaction are common biological tools. What is specific to this beetle group is the architecture that allows those chemicals to be stored separately, combined under pressure, and expelled in controlled pulses rather than in a single uncontrolled release. The pulsing, it turns out, is important. A continuous release of the same chemical mixture would overheat the reaction chamber and potentially damage the beetle itself. The pulsed delivery allows the chamber to cool slightly between pulses, which keeps the temperature at the exit nozzle within a range the beetle can sustain without self-injury. The beetle has essentially solved a problem in combustion engineering that human designers working on pulse jets encountered separately and much later. There are roughly 500 known species of bombardier beetle, distributed across most of the world's temperate and tropical regions, living in soil, under stones, in rotting wood, and along stream banks, quiet, ground-level animals spending most of their time moving slowly through the dark material of the forest floor and doing nothing that would distinguish them from any of the thousands of other small beetles occupying the same habitat. The spray is not something they deploy continuously. Most of their time is spent walking, foraging, and sheltering, and the chemical defense is reserved for the specific moments when it becomes relevant. A bombardier beetle crossing a patch of bare soil on a warm evening is, for all visible purposes, a small brown beetle crossing a patch of bare soil, which is not something that draws particular attention. The interior of the animal is its own business. The archerfish lives in mangrove channels and brackish estuaries across Southeast Asia and northern Australia, in the narrow zone where the root systems of mangrove trees descend into tidal water and terrestrial insects move along stems and leaf surfaces, a meter or two or three above the waterline. The fish hunts those insects by spitting water, a description that understates considerably what is actually involved. The water jet is shaped by a groove in the roof of the fish's mouth, formed when the tongue is pressed upward, and the jet is given additional force by the closing of the gill covers. It is precisely aimed, which presents a problem that is worth sitting with for a moment. The archerfish looks up at an insect through a surface that bends light. Refraction at the water-air interface displaces the apparent position of any object above the surface from its actual position. A ray of light entering the water from above bends toward the vertical as it crosses the interface, which means the insect the fish sees is not in the direction the fish's eye calculates it to be in. If the fish uses any simple geometric rule, despite this, archerfish hit their targets with high accuracy. Research has suggested that the fish compensates for refraction not through a general learned rule, but through direct individual experience with the specific viewing angles and water surface conditions it encounters in its particular habitat. The more involved finding came from experiments tracking the fish's jet trajectory against moving targets. An archerfish watching an insect walking along a stem above the waterline does not aim the jet at the insect's current position. It adjusts the trajectory to intercept the insect at the point where the insect will be when the jet arrives, which requires the fish to estimate the insect's speed and direction of movement, compute a future position, and arc the jet to that position, rather than toward where the prey is. This is not a behavior that can be explained by a simple reflex directed at a visual stimulus. The computation involved, whatever form it takes in a fish's nervous system, is genuinely predictive. It models a future state of the world and acts on that model rather than on what is directly visible. The insect falls into the water. The fish takes it from the surface. The whole process, from the fish's initial sighting of the insect to the insect's impact with the water, takes a few seconds at most, and most of that time is the insect walking along the stem, unaware. Archerfish live in groups, and there is a social dimension to their hunting that adds another layer to the picture. When one fish fires a jet and knocks an insect from its perch above the water, other fish in the group often rush toward the impact point. The fish that fires the shot does not always retrieve the prey. Nearby fish, which may not have been watching the insect at all, appear to respond to the water disturbance at the surface when the prey lands, and those fish can outswim the shooter to the food. Experiments have found that non-shooting fish in a group will, over time, learn to predict the direction of a prey item's fall from watching the angle of a shooting fish's posture before the shot is fired. They read the aim of their group member and position themselves to intercept the falling prey. The fish that shoots has, in effect, provided information to its group that the group uses against the shooter's own interests. The social dynamics this produces have not been fully worked out. The picture that has emerged from the research is complicated and continues to get more complicated as more experiments are run. The Omoctopus mimicus was first collected from Indonesian waters in the 1980s by divers who noticed that a particular octopus seemed to be doing something unusual. It was not until 2001 that the species was formally described in the scientific literature, and the behavior that had attracted initial attention was also formally documented. This octopus, now called the mimic octopus, alters its entire body posture and pattern of movement to impersonate specific other animals. The impersonations are not vague or approximate. When mimicking a flatfish, the octopus gathers all 8 arms into a compressed horizontal disc shape, ripples the skin along the disc's margins, and moves across the substrate with the characteristic undulating motion of a flatfish swimming low over the sand. When mimicking a lionfish, it holds its arms spread above its body in the pattern of lionfish fins, allowing them to drift in the current in the way that extended fin rays drift. When mimicking a banded sea snake, it presses most of its arms into a crevice or burrow and extends 1 or 2 arms in an alternating dark and pale banded pattern, moving them with the slow sinusoidal motion of a snake. The selection of which animal to mimic appears not to be random. Field observations have found that the identity of the specific predator present at the time influences which impersonation the mimic octopus adopts. When damselfish are the threatening presence, damselfish that, in the wild, are known to avoid banded sea snakes, the octopus tends to adopt the sea snake posture. The choice, in other words, seems to respond to the specific threat, not just to the presence of threat in general. What cognitive process underlies this selection is not settled. Whether the octopus has a representation of the predator's identity and a corresponding representation of which impersonation will be most effective, or whether the response is more directly conditioned through experience with specific predator types in its specific home range, or something else, the research does not reach far enough in to answer the question cleanly. What it does establish is that the behavior is flexible and contextual in a way that distinguishes it from most known examples of fixed-form mimicry in other animals. The mimic octopus lives on sandy reef flats, often in water that is relatively shallow and exposed, moving across open substrate in a way that leaves it visible from multiple directions simultaneously. It is not a small animal relative to other reef flat species. A mature individual can extend across a span of roughly 60 centimeters, including the arms, but it is soft-bodied and without any physical defense, more substantial than its skin. The impersonations it produces are its primary protection in this exposed environment, and they are produced quickly, fluidly, without any visible period of deliberate preparation. The animal moves across the sand in its resting form, and then the form changes. The arms gather or spread, the skin shifts through patterns, the movement style alters, and something that was an octopus becomes something that reads, at a distance and in the context of the reef, as a different kind of animal entirely. When the threat passes, the resting form returns. The octopus continues across the sand as itself, arms trailing loosely behind it, skin cycling through its ordinary resting colors, grays, browns, ochres, the shifting textures of sand and shallow water. What ties all of these animals together is not simply that their capabilities are extreme by some general measure, but that in each case, the capability is embedded in an ordinary life, a life of foraging, resting, maintaining territory or burrow or social position, of moving through a specific environment shaped by the pressures of that environment over time. The mantis shrimp spends most of its hours in a burrow. The electric eel drifts slowly through dark water, most of its discharges too low in voltage to notice. The bombardier beetle walks through leaf litter, its chemical chamber inert. The archerfish hovers below the surface of a mangrove channel, watching. The mimic octopus moves across the sand between episodes of impersonation, an ordinary cephalopod going about its ordinary day. The capabilities exist alongside all of this, present but not constantly deployed, available when the specific moment requires them, and folded quietly back into the general background of the animal's life when the moment passes. There is a sense in which the reef flat, the mangrove channel, the murky river, and the forest floor are not the dramatic stages they are sometimes made to appear. They are simply places where specific animals live, each shaped over long periods by the specific pressures of that place, carrying whatever it carries: club appendages, electric organs, chemical chambers, jet anatomy, a flexible repertoire of postures, as the ordinary equipment of its life. The capabilities that attract attention from outside are, from the animal's perspective, to the degree that perspective is a meaningful term here, simply what its body does. The mantis shrimp does not experience its own strike as extraordinary. The archerfish does not experience the compensated trajectory of its jet as a solution to a geometric problem. The things they do are the things they do, woven into the texture of days spent feeding, sheltering, reproducing, and occupying the particular portion of the world that is theirs. The mimic octopus on its reef flat holds the sea snake posture for the duration of a threat, arms pressed to one side, skin striped pale and dark, one arm extended and moving slowly. The damselfish that had been approaching the flat from the left turns and moves away. For a moment the octopus holds the posture, the arms still, the skin still banded, and then, in a way that is unhurried and seems to require no particular effort, the posture releases, the arms spread and trail. The banding fades through a brief intermediate pattern of pale dots and mottled gray into something warmer and less defined. The octopus moves away from the burrow it had been approaching, crossing a stretch of open sand toward the next patch of rubble, its arms drawing across the substrate in the loose, fluid manner of an octopus not currently impersonating anything. The water above it is shallow enough that the late light from the surface reaches the sand in wavering bands, and the shifting skin of the animal catches those bands in passing, briefly lit and then not, moving through the shallows of a tropical reef, flat in the same unhurried way it has been moving for whatever portion of its roughly 1 year lifespan has already elapsed. The reef flat continues around it. Somewhere in the rubble nearby, there are pistol shrimp in burrows they have built and maintained through weeks of careful excavation, each one a centimeter or two longer than a human thumb, resting in the sleeping chambers they share with the gobies that serve as their eyes on the world above. Somewhere in the water column a few meters out from the flat, the continuous crackling of a dense population rises from the sediment and spreads outward in all directions, a sound as ambient as wind, present through the day and through the night, produced by animals most of the fish swimming through it have no reason to notice. Somewhere in the dark water beyond the reef, a mantis shrimp rests in its burrow, its dactyl clubs folded inward, its elaborate eyes closed, its body doing whatever the body of a mantis shrimp does in the hours when it is not hunting or defending. In a river system on another continent, in water that no light from the surface reaches clearly, an electric eel moves slowly through the space between two submerged roots, the weak continuous field of its navigation sense spreading outward, a body length in all directions, registering the shapes of things, roots, substrate, the still water itself, as slight distortions in the electrical geometry of its immediate world. The forest floor in a temperate woodland somewhere holds a bombardier beetle under a stone, its chemical chambers full, its body still, its legs folded in the specific configuration of a small ground beetle resting in the dark. It has not used its spray in some time. It may not use it again for days. The stone above it sits on damp soil, and the soil holds the mycelium of fungi and the galleries of ants and the slow movements of earthworms drawing leaf material downward, and above the stone the leaf litter holds moisture from the last rain, and above the leaf litter the trees hold their canopy against the sky, and the sky above the trees is moving into the long blue hour before full dark. The beetle rests below all of it, warm enough, still, doing nothing in particular, which is to say, doing what most animals spend most of their time doing, which is simply continuing. In a mangrove channel somewhere in the islands of the Indo-Pacific, an archerfish rests below the surface, in water that is warm and slightly brackish, watching the stems above the waterline with the particular quality of attention that a fish hunting from below has had a very long time to develop. A small insect moves along a stem. The fish tracks it